All right, folks. So I wanted to pick up right when we left off from this week on Chapter 8. Just a second. The integral proteins that we see here, right? So those can actually vary depending on what this is. So they can vary, right? And sometimes the carrier molecules that we're talking about here can be used to identify a bacteria. And one of which we're going to look at in this course is the oxidase test. We actually can use this test to identify if this particular organism has an electron carrier called cytochrome C. And if cytochrome C oxidase, which is the enzyme used for this process to work, this electron carrier, if it's present, right? If it's present, the oxidase test can test for that, and thereby we can figure out again what it might be. All right, so for example, Neisseria gonorrhea, which is the infection that causes gonorrhea, right? In a living organism like a human, they actually use cytochrome C oxidase, that same enzyme that we're discussing, right? And so when we use this test, we know that the gonorrhea, for example, is oxidase positive. And that's one way we can tell, for example, this is in fact this organism in question. So we can use even the types of electron carriers in the electron transport chain to identify an unknown species, right? Salmonella is the opposite, right? Okay, salmonella causes foodborne illness, but it does not have cytochrome C oxidase, and so we would use that test, and that test would be oxidase negative, right, versus positive. Again, keep that in mind because we'll use this biochemical test in some of our lab. Okay. All right, so just to kind of, before we kind of move on from here, looking at the actual process of the electron transport chain, the last step uses a process called chemoosmosis, right? Chemoosmosis, which if you divide this, or chemiosmosis, where if you divide this in half, that's actually two words. This means chemical, right, or chemistry, and osmosis means what? Permeability, right, or allowing stuff to go through, right? Osmosis is the movement of certain things from one area to another area through a semi-permeable membrane, right? And in fact, this is what we see in the last part of the electron transport chain, where we see these molecules traveling from one side of the membrane to another side of the membrane, back and forth. And when they do that, when these hydrogen atoms get pumped back at the very last step, it actually initiates and synthesizes the phosphorylation reaction that recharges ADP to ATP, right? So every single time, for example, if this was the plasma membrane and we had a bunch of hydrogen atoms here, and we had what we call a proton pump, every single time one hydrogen atom goes past the plasma membrane, we're taking ADP and a phosphate group and making ATP. That's what we talk about when we talk about the phosphorylation reaction. In fact, is what we're discussing here, the process of recharging ADP into ATP. All right, and we talk about this being this process here, chemoosmosis. Okay. All right. So again, just this kind of recaps chemoosmosis, what we're talking about in the electron transport chain, where we have the electron carriers dropping off the electrons, redox reactions. We go from a high energy state to a lower energy state. The energy released is used to pump protons across the membrane and then again recharge ADP into ATP, right? Which is our end goal. And again, 34 is what you see in the electron transport chain. All right, so this is our proton pump. We call this a proton pump because these, in fact, are protons, right? Positively charged molecules, right? Protons. And in fact, they go, as you see here in the arrow, straight through and from one side of the plasma membrane into the other side. And as they go through, in fact, that proton pump changes shape and then charges ADP and a phosphate group and makes ATP. Essentially, that's the end stage for producing all of those 34 ATP that we see in the electron transport. Okay, again, the process of moving protons from one side to another is chemiosmosis, and before they move, we call that the proton motive force, where all the hydrogen ions are on one side of the plasma membrane. The enzyme that we discussed, that's the proton pump, is called ATP synthase. Remember, ACE is an enzyme. The protons go from a high concentration to low concentration, as is required in osmosis or diffusion. In fact, we go from a high concentration to low concentration. ATP synthase is the one that captures energy from flowing protons, and it uses that energy to stick into ADP and another phosphate group to then become ATP. And so we see this, right, for each FADH2 electron carrier, we make an average of 2 ATP molecules, and for an NAD, you make an average of 3 made there. And again, in a eukaryotic organism, just to kind of recap, this is in this part of the mitochondria we call the cristae. That's the inner mitochondrial membrane. And in a prokaryote, it's the plasma membrane. All right, so talking about the final electron acceptor, we know that an aerobic respirator, which is a fancy word to say an organism that uses the oxygen as a final electron acceptor in cellular respiration, right, will use O2. In an anaerobic organism, they do not require O2, right, as the final electron acceptor. Now, notice that they can use other things other than oxygen to actually be the final electron acceptor, because they're going to need a final electron acceptor. It's just that anaerobic organisms don't require oxygen to do that. And so here are some examples in this table here that show final electron acceptors for anaerobic respiratory chains, or the electron transport chain for these particular organisms, right? So if you're a nitrogen fixer, which you're a bacteria that actually converts nitrogen, you actually use nitrate. This is the chemical, right? NO3, nitrate, as the final electron acceptor. And when you bind it to the electron, it gets reduced into nitrogen gas. Nitrogen gas. Okay? Instead of water being released, it'd be nitrogen gas that is released from this type of bacteria that is what we call a nitrogen fixer. Now, how is this relevant to some of these things? Where do you see this in the real world, right? A lot of the bacteria, for example, that you find in soil are what we call nitrogen fixers or fixators. And in fact, they take nitrate, okay, from decomposed, let's say, plant life that's dead. They'll actually decompose that or decompose deceased living organisms, right? They'll actually use that nitrate as their final electron acceptor and convert it into nitrogen gas. And nitrogen gas is important because that actually goes into the air and it also stays in soil. And nitrogen, if you know a little bit about gardening or plants, nitrogen is one of the crucial ingredients to actually have good plant growth, right? So we call this nitrogen fixation. Methanogens, which are organisms that actually make methane, they actually use carbonate, right? Carbonate gets used as the final electron acceptor, and they convert it into a chemical that we call methane, right? Or methane gas. And in fact, there are some organisms that will do this. For example, in the GI tract of organisms. Humans are those examples, but agricultural animals can be examples of that. For example, cows, when cows eat grass and what have you, it's the bacteria inside of their GI tract that converts carbonate into methane, and that methane comes out of, you know, the other side of things, right, as a methane gas. Okay. We also have methane in the ground, by the way. So if you know anything about oil and gas, as you're drilling for oil, you hit these pockets of this gas, this methane gas, and it's really from organisms that have broken down organic molecules and are using carbonate to convert as a final electron acceptor to get energy, and they convert it into methane gas. And those pockets of gas are found in the ground, trapped. And when we break open that area, sometimes those trapped pockets of methane get released. And if you've been in West Texas and you've seen all the, you know, the oil being pumped, you also know that there's always gas being burned off. They light the gas because the gas that's released into the air just naturally will cause environmental changes, so they rather burn it, right? So that's methane gas, right? And then you also have sulfate reducing organisms that will actually use sulfate as a final electron acceptor, and they will reduce that'll become hydrogen sulfide. Hydrogen sulfide. Okay, so just a little note here. Aerobic respiration versus fermentation. We haven't talked about fermentation, but both are anaerobic. They don't use oxygen, all right? Fermentation doesn't use the respiratory chain. Obviously, it doesn't go all the way to an electron transport chain. Respiration does. Cellular respiration does. Fermentation occurs in the cytoplasm, and the electron transport chain is always with the membrane, right? Whether that be a plasma membrane or the cristae of the mitochondria, right? So just kind of comparing aerobic respiration versus fermentation. Okay, so looking at your production of ATP here, right? Theoretically, you can make 38 ATP from one molecule of glucose, right? That's that C6H12O6 molecule. Theoretically, you can make 38, right? And why 38 instead of 34? We just talked about 34. Well, 34 are really for just the electron transport chain, but we haven't included the two that we make from the Krebs cycle and the net two that we make from glycolysis. So if you add all of these together, then that equals to your 38 there, right? 38 there. And again, this graph, this this table just shows you, again, if you go all the way to the left, how many we invest, right? And we know in glycolysis you use 2, right? We put in 2 ATP molecules. How much you make total, right? You actually make 4 in glycolysis. So you actually make 40, but you net, again, net 38, because you use 2 up, right? Again, keep that in mind. Don't forget that. Don't confuse that. Okay, so we can summarize the reaction in this following manner, right? Here's the glucose molecule. We need some oxygen. I didn't mention that, but we need a little bit of oxygen. Again, I did actually mention that because this is used as the final electron acceptor. And again, this is aerobic, right? In theory, you need 38 ADP there. You need these 38 phosphate groups, and they're actually binded this way. Phosphate's not by itself, and I didn't mention that. It's actually looks like this here, but you need those obviously because you need to make then the ATP that you need to make. You'll make 6 molecules of water and 6 molecules of carbon dioxide, right? And we know where these come from other steps in the cellular respiration cycle. We know that this comes from the electron transport chain, and this is made in the electron transport chain, right? So these get made in the electron transport chain. These get made in other places, right? The intermediate step, Krebs, is where you find those. Okay. So what we're going to look at here very quickly are other pathways for catabolizing nutrients, right? Okay, other ways that that organisms can actually do this, right? Break down other things, right? Okay, so glycolysis is not the only pathway, in fact, that we see in living organisms, right? Okay, so again, glycolysis is the most preferential. That's why you see it the most common in most cells, but there are some organisms like bacteria that actually have alternates, okay? This is one of the alternates. We call this the pentose phosphate pathway, right? Pentose phosphate pathway. And actually, instead of using glucose as the molecule that you want to break down, we use another type or other types of carbohydrates. We can use pentose, which are 5 carbon sugars. And we can break those into 3 triose, which are 3 carbon sugars. And then we can use those to build 6 carbon sugars that can be used in glycolysis. So this is telling you that glycolysis can still be there. It can still exist in a organism. What we're seeing here, and the reason why we're seeing this as an alternate pathway, is that sometimes an organism cannot get glucose. They cannot harness glucose in the environment. They can't get it. Might be a change in the environment. That nutrient is not there. So because of that, they have engineered this over time because of evolution that they can actually take other sugars other than glucose, like pentose, and still convert it into a 6 carbon sugar, and still take that 6 carbon sugar and use glycolysis, the process of glycolysis. Remember, the process of glycolysis can only start because it has the enzymes made to convert those steps, that 10-step process. Okay? So it can also do some really interesting things. It can actually take glucose 6-phosphate, which is part of glycolysis, one of the steps of glycolysis. So once glycolysis gets made, gets started, it can take the part where it forms this glucose 6-phosphate, and it can actually turn it into a pentose, a 5 carbon sugar, And that can be used to make nucleotides if it wants to, and amino acids. So why would you want to make nucleotides and amino acids? Well, it's because you need to, right? The organism might need amino acids at that time to make proteins, or it might need to use nucleotides to make more ATP molecules, or to make more DNA or RNA. In any event, the really awesome thing about these types of pathways is that you can take intermediates, little middle points between these steps that we've been discussing, and then take them into other places to convert them into other macromolecules, right? So, in fact, the pentose phosphate pathway gets used in many bacteria. Bacillus subtilis uses this pretty frequently. Even E. coli uses this, and other bacteria, as a way to harness and make ATP in an alternate pathway. We call this pentose phosphate. Okay. What we call the Enitter-Dordorf pathway, sometimes it's known as the ED pathway for short, but the ED pathway actually uses glucose, right? And it makes one molecule of ATP per glucose. And it's actually a broken down, simpler version of this process that we had talked about. And it also makes electron carriers that also make some energy as well. And again, this we think is the pre-pathway. This is the original pathway before cellular respiration was invented. Okay. Before cellular respiration, scientists, we believe that the ED pathway was started first, in fact. So where do you find the ED pathway? You find it in gram-negative bacteria, those ones that are obligate, or aerobes or aerobic organisms. Okay. So what happens when an organism doesn't have a final electron acceptor, right? So that's a common occurrence, by the way, in an organism. Let's say a bacteria lacks a final electron acceptor, right? If it, for example, doesn't have oxygen, or if it doesn't have nitrate, right? Or if it doesn't have, any of those types of other final electron acceptors. So if it doesn't have that, some organisms elect, or some of them have an alternate pathway to break down nutrients. One of those pathways is fermentation, right? So again, if an organism doesn't have a final electron acceptor, if it's lacking one, or sometimes some organisms don't even have the electron transport chain, they use another pathway, we call fermentation. Okay? So here's the goal of fermentation. It's not really to make ATP, right? We don't use it. Fermentation doesn't really, its main goal is not production of ATP. It's actually to sustain ATP production by glycolysis when the respiratory chain is not available or doesn't exist. Okay? It's just to continue the process of glycolysis. Yes, you need ATP, but that's not its main goal. It's just to continue glycolysis. Because glycolysis and some of their pieces can be used for other things, and those other processes can actually make energy for the organism. But glycolysis is the first step in any type of metabolism, any type of energy production for metabolism. And because of that, the main goal of fermentation is not ATP, but actually continue the process of glycolysis. So in fermentation, we do have electron carriers, and actually it's NADH. And NADH, and its goal is not to pass the electrons to the electron transport chain, because that's not what exists here or is not available. It actually passes its electrons to organic molecules like pyruvic acid, which is the end step of glycolysis, right? NADH is restored, so it gave off its electron in the hydrogen group, so it becomes NAD plus, and so it picks up more electrons, and then glycolysis continues and continues. Okay? So here's what we see, right? Here's pyruvic acid. Okay? So again, the process of fermentation produces side products, right? One of them being pyruvic acid. So that's its main goal, right? To continue to keep glycolysis moving, right? And glycolysis produces then pyruvic acid for the organism. In the process of fermentation, that pyruvic acid, in fact, depending on what type of fermentation process, can be used to make ATP for the organism. Now, when it goes through this process, depending on which fermentation it is, in the process of making ATP, it also makes a side product. And here are some of the examples of the side product. Okay? Side products like lactic acid, ethanol, and some other acids, and butadienol. Okay? Which you can actually detect if this type of fermentation occurs in what we call the VP test. And many of you will actually inoculate an MRVP test this week. And whatever organism that you're inoculating the MRVP test tube with and have growth will test positive, can test positive for this type of fermentation. Okay? But stepping back, fermentation, again, its main goal is to keep glycolysis moving, but glycolysis also will produce, by moving glycolysis, produces pyruvic acid, and depending on which organism undergoes fermentation, we see the side products being. Okay? So, fermentation does also occur in eukaryotic cells, definitely in prokaryotic cells, but you see that in eukaryotic cells. One of them is yeast, for example. Yeast has the fermentation process, right? And the pathways are named by the end products, right? That's what we just saw, the end products there. Fermentation can also not only catabolize carbohydrates, but it can also catabolize other nutrients. So they can actually break down amino acids, fatty acids, like your proteins and your lipids, for example, can break those. So these here, right, the ones that you saw in the figure 2 slides ago, are common types of carbohydrate fermentations. So you see those there, right? And again, they're named based on the products that they make. So if lactic acid fermentation produces lactic acid, alcohol fermentation produces alcohol, Mixed acid fermentation produces ethanol, alcohol, and other acids, right? And butanediol fermentation produces butanediol. Okay. Here are the two types of lactic acid fermentation. And we don't really need to know that, but this is the other two types of acid fermentation. But just to kind of give you an example give you an example of a type of lactic acid fermentation that we call homolactic fermentation. This right here, right? Yogurt, right? So the active ingredients, the active live cultures, in fact, of yogurt include Lactobacillus bulgarius and Streptococcus thermophiles, and those two of which actually produce, because of fermentation, produce lactic acid, and then lactic acid then actually breaks down and converts the proteins and the sugars in milk and converts it from milk into yogurt. Sauerkraut gets made in the same way, lactic acid fermentation, homolactic fermentation, and even our muscle cells in the human body can actually undergo this as well. And they actually, muscle cells use this type of fermentation first before they actually do cellular respiration. And when they do that, and if you undergo muscle cell movement a lot and build up the fermentation process, in fact, you build up lactic acid. And lactic acid is the one that actually makes you, the next day or the next couple days, depending, it actually makes you achy or sore. It's actually the lactic acid buildup. Actually, that's where the cramps also come from as well. Heterolactic fermentation is a type of lactic acid fermentation as well. And again, a lot of bacteria and fungi actually do this. And you can see that there. Alcohol fermentation. Okay. Certain bacteria do that, fungi, plant cells, but especially this right here, right? Saccharomyces cerevisiae, and that's actually a certain type of yeast, right? That yeast is the one that actually converts that grain, for example, and grapes into those products there, right? Mixed acid fermentation will produce many different types of acid and gases: formic acid, acetic acid, succinic acid, ethanol, hydrogen gas, carbon dioxide. All of these are what we call mixed acid fermentation. And usually your enteric bacteria, which is a fancy word to say your bacteria of your what? GI tract, right? And your colon, in fact. These types of bacteria actually undergo mixed acid fermentation. And for example, this is where you see gas buildup. Right? In your GI tract. That comes from the bacteria undergoing mixed acid fermentation. Okay. And again, I mentioned butanediol. Butanediol, you know, it actually converts that to butanediol. Okay. All right, so I just want to talk really quick on these, right? So just to mention for you that organisms can also break down lipids, proteins, and nucleic acids. So that's not only just carbohydrates. Organisms can actually break down these as well, other macromolecules, right? All right, so we see some of these, right? Bacteria can secrete these exozymes, which means they actually secrete enzymes out of themselves in the environment, and their goal is to break down the large macromolecules. Lipases, for example, which breaks down lipids, right, into smaller monomers, right? Proteases break down proteins. Peptidase breaks down peptides, and they break them down to their monomers, which are amino acids. nucleases can break down nucleic acids into nucleotides. And again, depending on the species, different species can actually have different exoenzymes. So they can have, you know, any of these or all of these, right? So, so this is figure, actually I don't know what figure this is, but this is a figure that you should see in your book, in fact. And this figure here is a really important figure to look at because it kind of ties a lot of things that that we've been discussing, right? One of them being the process of cellular, right, respiration. Which is glycolysis, the intermediate step, Krebs cycle. And we haven't included the electron transport chain, but those previous steps. And look what we see here, right? Look what we see down on the right side. We see that through the process of cellular respiration, those steps, some of the products or the middle products between glucose all the way down to all your electron carriers, all those middle steps, some of those can be brought in and made into carbohydrates. Amino acids can be made into lipids, and the Krebs cycle here can be made into amino acids. So we can take middle steps between all of the cycles and can convert them, or organisms can convert them back into things that they need at a given time. And that's what we see there. So it's interchangeable. Some of the products can be taken straight out, used to make different things. And in the same regard, this can actually work in the opposite way. We can take amino acids and stick them in these steps, and we would still have the process continue on, right? And that's the end goal, for the process to continue on. So it can go back and forth, in fact. So I'll draw the arrow this way, because in fact, it can go both ways. That's what we're seeing here in this. Okay, so we have to, you know, we don't have to, but what we like to look at is actually looking at diagnosing a particular pathogen. Again, if this is a clinical setting, if you have an infection, if you've got a patient that is infected and you want to diagnose the patient correctly and then treat the patient, one way we can do it is look at their metabolic properties, look at the type of metabolism they're undergoing, and we figured out through science and research that we can actually test the enzymes, right? We can test the exoenzymes that a certain organism, like a bacteria, will secrete, right, in the environment. And because we can test on those, then we can diagnose, okay, this bacteria is a species of, let's say it's E. coli, because I have certain types of enzymes that will cause certain changes on certain media, right, that we grow the bacteria on. And that's really what we're seeing here, right? Each of these pictures represents some of the media and some of the changes that we can actually read or look at because that certain species is secreting exoenzymes to break down certain types of macromolecules. And you notice here, for example, on the plate, this side is yellow or kind of a brown color, and this is blue. That's indicative of an exoenzyme that this organism has been secreting out. And we see this one here, where B, for example, is a certain color, A is a certain color, and then you see this here and this too, right? Same color, but what's the difference here? This one is looking at the agar staying in place and not moving. And look at this, right? This agar has become from a solid to a liquid, right? And we're looking at that change there too, right? Which means it's actually done. Okay. All right, so I'm going to go ahead and move on through all of this because this is talking about these types of catabolism. Same thing with this. This is anabolic reaction. So in the same regard, if we are breaking down stuff, organisms can also build up things, right? Okay. The process of gluconeogenesis, by the way, is actually building sugars like glucose from non-sugar material, right? And actually, you can go backwards, right? So again, we can actually build up, make glucose from other things other than carbohydrates, which is really neat to look at, right? All right. So sometimes we use things like glucose to build stuff like glycogen, which is a storage molecule for glucose. You see each one of these is a glucose molecule, kind of like a branch. And we can also use, for example, glucose to make these sugars like peptidoglycan. And if you remember this makes part of the cell wall, right, of an organism. Sometimes we can do this, and again, do not memorize this, do not study this, but all I'm showing you here is that some of the intermediate steps of glycolysis and the intermediate step here, sometimes we can take the products, side products from there, or the products between these steps, and we can take them, and then an organism can add them together like they're doing here, and they can make a lipid if they need to, right? This is, looks to be probably a triglyceride, right here. And again, there are essential amino acids. There's actually 20 essential amino acids. And actually we can take amino acids and build those up also. The organism needs it. Again, can use any of those steps to build amino acids, right? And in fact, we can build up and make nucleic acids if we need to. And again, look at the same process here, right? Purines and pyrimidines, right? Your A's and T's and your C's and G's. Or U if you're RNA, right? We use these to make DNA and RNA, right? So if we need those things, then we can use steps in the respiration to do this, right? Okay. Pathways that simultaneously function in anabolism and catabolism are called apibolic pathways, which means they can go back and forth. You can break down, and then you can also build up. So you can do this at the same time, or do one and then the other, and do one again and the other. We call this apibolic pathway. Okay? We're going to talk about this here as well, right? We can categorize organisms according to their nutritional patterns and how they make ATP. Where do they get those sources? Right? So if you're an autotroph, you can make organic carbon molecules from inorganic molecules, right? We call this carbon fixation. You can make organic carbon molecules from inorganic materials. Okay? That means you're an autotroph. Okay, if you're a heterotroph, in fact, you require an external source or an organic carbon. You need them. You can't make them on your own. You need an organic carbon molecule, right? That's what we mean by. So these talk about electrons, right? That we need to jumpstart to make the redox reaction, to kick off the redox reaction. If you're a lithotrope, you get your electrons, if you will, from inorganic sources. Okay? If you're an organotroph, you get them from organic sources. Okay? If you're a phototroph, you can actually use energy from light to make ATP. If you're a chemotroph, you use the energy from the chemical bonds that you break open to make ATP. Mixotrophs can actually do both, right? Again, the keyword phototroph, light, the chemical bonds. If, again, I'm breaking down carbon like I'm doing here, breaking down carbon, for example, like catabolic catabolism, cellular respiration, then I'm a chemotroph, right? So who's a phototroph, right? Harvesting energy from the light, right? So if you guess this, then that's absolutely correct. Plants actually use light to make ATP. But also there are some bacteria that do this too. There are some bacteria that actually can use light to actually make energy. And you would write that there. Or if you use light and you don't make organic carbon, you'd be a photoorganoheterotroph, and so on and so forth, right? And that's how you can actually name all of these here. Okay, as I mentioned, there's many tests to actually identify unknown bacteria. And because species have a lot of different metabolistic profiles, they actually do different types of metabolism for different types of macromolecules. Then if we know that to be true and have studied each one, then this would be a way to identify a microbe. And in fact, look at this, there's over 1,000 different tests to identify microbes. 1,000. So it's really neat to look at. Sometimes you grow these, sometimes you actually add chemicals to their growth, sometimes it's genetic, sometimes it's metabolism, but everything leads to the metabolistic property, right? So again, you can use these many tests to identify that, right? Okay. So again, just to kind of go back to this, I just want to kind of touch on this because this is important not only in lecture, but when we look at labs, right? If we take an unknown sample, if you want to identify it, you make a pure culture. That's what we've been practicing on to actually, you know, subculture something so you can identify it as a pure culture, right? Then you use stains, a microscope to look at the organism. And then you take that culture itself through testing. You do many of these metabolic tests or biochemical tests to further identify the organism. And this is the process. Doesn't matter if it's a microbiology lab course in college like you're taking, or if you're in a hospital setting trying to identify an unknown pathogen, you would work at it the same way. Identify it, isolate it as a pure culture, stain it, run it under a microscope, run through some tests, and then again you have bacteria, right? So here's some of the tests. You don't need to memorize these tests. I'm not going to put these on an exam here, but we're all going to use some of these in the lab, right? So there's one that tests for the breakdown, remember this catabolism, the breakdown of an amino acid or amino acids. So again, you can use this test to see if an organism breaks down amino acids. There's one that uses fermentation. That's the one we're working on this week. Actually putting bacteria in there, watching it grow and seeing a change. So if organisms can undergo fermentation of a certain type of carbohydrate, then this fermentation process will indicate that. This one here, MRVP test, you use this test to actually look for mixed acid fermentation and butanediol fermentation, those two fermentation processes. And if an organism can do both of those, then you would test for those, and again you would see the change there. Okay, an oxidase test is a test to test for that cytochrome C oxidase, which is an enzyme that an organism uses for the electron transport chain, right? And if the catalase test uses then test for the enzyme catalase, and that's an enzyme that uses to break down a toxic substance that we call hydrogen peroxide, right? Which is a normal process of metabolism. We see this occurring as a side product, as toxic in an organism that can have an enzyme that breaks that down. Have that there as well. And finally, you use this strip, right? This strip here, we're not going to use, but you can use this in a hospital setting. And in fact, each of these vials contains a certain test, certain biochemical test. And when you put the organism into there and identify, again, depending on what color, what indicator is positive or negative, and how many there are, and then you can actually look at it. Its color, it's coded with a number, and then you can identify that number. And this is really how that works in a hospital. All right. So kind of, again, this is the last part of it, but again, at the end of the chapter, there's always the end that kind of summarizes a lot of the main points that we talked about in this PowerPoint. All right, folks.